Pleasure to talk about the new concept, the Holobiont imperative, why our microbes are key to our health. What I'm going to talk about is the microbiome, which we consider all of the host-associated microbes. We consider organisms as meta-organisms, and that means all these microbes together with the host. And I would like to remind you that we know that humans contain more bacteria cells than human cells, that we have about 150 times more bacterial genes in our body than are in our genome. And I would like to show you that microbes have a major impact on biology and fitness. And I will do that in 7 very short chapters. I give you an introduction. Then I talk about functional aspects and the role of the microbes in immunity. I talk about microbial colonization, about microbe-neuron interactions. And then I will tell you that the partnership between the microbes and the host is in danger, particularly these days because of overuse of antibiotics. I will give you my view of what we can do. And I will not end this lecture without mentioning that the pandemic, the COVID-19 pandemic, has, of course, impact on the normal microbiome. With that, I give you a brief introduction. I would like to tell you that all what we know today is because of major advances in technology, and this is particularly sequencing technology. This is a few in our lab at Kiel University in Germany, where we sequence every day genomes, transcriptomes, metagenomes. And this allowed us to look deep into tissues and to discover that any human, animal, and plant tissue is indeed full of microbes. This is a wonderful picture here, which shows you from the Rob Knight lab that all the colorful spots indicate presence of microbes or microbial products in different parts of the body, the upper and the lower part of the hands and the foot and the head and the whole body, and means all organisms, humans, animals, and plants are covered. Here is the plants or the leaves or the roots with a biofilm of microbes which are beneficial. And if you remove them, the plants, the animals, and humans are getting sick. This is the major message. In nature, there are no germ-free organisms. All organisms are associated since their evolutionary beginning with microbes. For that reason, we consider microbiomes as the invisible component of our world. And this has dramatic consequences for human and animal health. And I would like to show that on a graph which shows you that in the last 70 years, numbers of diseases, asthma, diabetes, allergies, chronic inflammatory diseases, multiple sclerosis, adipositas are increasing with an enormous rate. Doctors see more and more of patients suffering from that. We call them environmental diseases because these diseases were not there not in that number 70 years ago. There is no prevention and no cure against any of these diseases. You also see that include neurodegenerative diseases. There is some improvement in the symptoms, and people give these diseases different names. Some people call them lifestyle disease. The WHO calls them non-communicable diseases because they think that these diseases cannot be transferred from one individual to another one. And an interesting paper in PNAS some years ago called them nature deficit disorders. And that actually brings it to the point because we think that all of these huge, in numbers, huge diseases, which are affecting all the societies around the world, have a disturbed host microbiome network, and we call it dysbiosis. This is a critical and a common risk factor for all these type of diseases. And we know that, among other aspects, from the fact that fecal microbial transplantation, the transfer of fecal material from so-called healthy donors into patients suffering from these diseases, in some cases, and sometimes, not always, leads to an improvement. And my colleagues have shown that in this fecal transfer material, this is nothing but microbial products, not really living microbes, but part of the microbial cell body, the cell wall, or most small microbial metabolites. Since this material can linder and improve the symptoms for some of these diseases, the question is justified: can microbes help? And by the way, this technology is nothing, is not new at all. The Chinese have invented that 4,000 years ago. It's called yellow soup, and the Chinese emperors have given that to drink to their soldiers when they were suffering on certain diseases. From all that we have a tight interaction between the microbes and the host, and that medicine and biology has focused in the last 100 years exclusively on the host when it wants to understand development, behavior, evolution, disease, and health. We have to expand that concept and include the associated microbes, the viruses, the symbionts, and for that reason, we consider organisms as meta-organisms or holobionts, and mean that any particular host is stable associated with all kinds of different microbes. And as I said before, if you remove them, you get sick. We do that in a research center here at Kiel University, which is called Origin and Function of Meta-Organisms. I'm sitting right here at the moment, to understand at a functional level, how do these microbes interact with the host and vice versa. In the second short chapter, I would like to give you one of the many functional aspects we have learned in the last few years, and maybe the most important aspect is that microbes contribute to our immune system. Please consider from now on microbes as a major part of your innate immune system. And the evidence is very obvious. If you do with any experimental model organism, an experiment where you remove all of the bacteria, we call that germ-free, and you can do that with my model system, which is the freshwater polyp hydra. You can do it with flies, you can do it with worms, you can do it with mice. We have a germ-free facility out there with 3,000 cages of germ-free mice. And then you can watch what happens in these animals when the bacteria are absent. And when you do that, in this case here, you can see that here a fungi is attacking the tissue in a deadly manner, and these polyps will simply die. In the presence of microbes, the polyps are completely healthy and fine. Only when we remove the microbes, fungi can attack. And the reason for that is that by removing microbes, we have removed the protective layer along the epithelium. The microbes are sitting in high diversity in a sugar-rich layer, so-called the glycocalyx, which in our gut is the mucus layer, and they interact with each other. Microbes talk to each other, and that is essential. And after that talking, they produce something, and here comes a fungi and a sporidium approaching the epithelium in this electron microscopy picture, and this talking of the microbes produces an antifungal molecule, and this antifungal molecule prevents that this fungi can attack the epithelium. In the absence or the disturbance of this microbiome, the fungi can attack, and then the animals are either heavily sick or just dying. The same happens in humans. If you come back from the dentist, because you have parodontitis and the dentist gave you oral antibiotic to eat. Then in your oral cavity, you have removed, of course, the bacteria. You have only removed the bacteria, not the fungi. And when the bacteria are gone, fungi can grow in our mouth. There is candida living all the time at low numbers. But if the bacteria are gone, the fungi can enormously increase their growth rate. They have open niches to colonize, and we have fungi infection, which we call thrush. You never have fungi infection and this type of ugly fungi grow in your mouth when you have a normal microbiome. But if you eat oral antibiotics, then the fungi can grow. So that means in normal cases, bacteria prevent fungi from growing. In the third chapter, I would like to show you how do we get the microbes in our body from the very beginning when we were newborn babies. And when we were newborn babies and the mother is delivering us, we get our first microbes from the vagina of the mother. And then these microbes are very important to train our immune system. And if the microbes are not there because the mother took too much antibiotic or had other issues, then the babies are born and the babies get different, for example, T-cell populations and a different immune system. And normally then the baby develops and after 1 year has a very healthy microbiome with also anti-inflammatory bacteria which help us in wound healing processes, for example. Now, if the baby is not born by vaginal delivery, but by cesarean section, all of this cannot happen, and the baby gets the microbiome from the birth room or from the architecture or somewhere else. This is essential and a problem because cesarean section babies have a very different microbiome than normal babies. And this different microbiome here indicated compared to the normal vaginal delivery babies makes these babies and children lifelong medically compromised. This is particularly important because the number of cesarean delivery is steadily increasing worldwide, with some countries about more than 50% of all the births is done by cesarean section. In the US, about 30%, and in Europe, about 25%. So cesarean sections are necessary in some cases, but not in all cases. And we do not do anything good to the baby if the baby does not get the microbes from the mother. In the next chapter, I would like to point to a very new field of research which deals with the interaction between the neurons and the microbes, or the microbes and the neurons. This is a fascinating new frontier, and in the literature you can find that as the so-called gut-brain axis. That means microbes, particularly in the gut, seem to have an influence on our central neural capacities, which has many implications, and we are just at the beginning of understanding what's going on. When we look in our model system, we realize that microbes, which are living in this mucus layer outside the epithelium, are in direct contact with sensory neurons, which are around in the epithelium, around the body column. And if you look into this picture here behind the scheme, you see here little red dots. These red dots are microbes. The green are the protrusions of the neurons, and you can even physically see that neurons can reach in them to the microbes and directly touch them, so to speak. That doesn't tell you that they do anything together, but that they could anatomically do something together. And the same happens in our gut or in our skin, where neurons are in contact with the microbiome. And we had the hypothesis that an animal and a human body, the nervous system, is actually built to enable the interactions with the symbiotic bacteria. And we could show that neurons, here is a typical ganglion neuron, transgenic, expressing GFP, that they produce small molecules, antimicrobial peptides, and with these peptides, they regulate the normal, the beneficial microbiome. So neurons, and these in these animals here are actually evolutionary, one of the oldest neurons we know of. These neurons shape the microbiome. So the nervous system participates in regulating the meta-organism by controlling the composition of the, and also the spatial localization of the colonizing microbes. So neurons communicate with peptides and other molecules with the microbes which are living on the epithelium. The next question then is, could bacteria directly influence the neural activity in the tissue? So does this interaction and communication goes in both ways? And for that, we look into a spontaneous activity of the enteric nervous system, the gut. This is an isolated gut from a mouse. It sits in a Ringer solution, and what you can see is they continuously contract and expand. They contract and expand, and this has nothing to do with food transportation, and we are just working on the why question, why they are doing this, but it is a spontaneous contraction. And in humans, here you can see again the small intestine. This is from a patient which has problems, peristalsis problems. And what you can see is that this movement goes back and forth, and that is causing pain. Something is not working properly in these body contractions. And the question we are asking, or we were asking, is who controls these spontaneous contractions? And we did it again in this model system, which contracts about 10 times regularly per hour. And if you disturb that, the animals are either sick or they die. You need these spontaneous contractions. And we measured the number, the amplitude, the regularity of these contractions, etc. And what we could show is that in germ-free animals, the number of contractions is drastically reduced. And if you give back the normal set of microbes, then these contractions come back. You can rescue that problem, the absence of microbes, nearly completely. That means microbes have an impact on these spontaneous contractions. Our model currently is that a diverse microbiome outside this gut-like tissue produces something, a small molecule. This molecule activates a receptor. This receptor on a nerve cell, and this activated nerve cell activates the muscles. The question is, what type of receptors are these? And we went back to the patients which suffer from gut dysmotility, and in particular from the irritable bowel syndrome. And we took genes from GWAS, whole genome analysis, to ask what type of receptors are present on pacemaker cells, and could these receptors interact with the microbiome. And our model shows you that there is numbers of iron receptors, but also receptors from the immune system, which interact with the microbiome from early animals to the mouse and the human, and thereby control these spontaneous interactions. All of that tells you that the nervous system emerged in evolutionary times to orchestrate multiple functions, sensory input, motor output, of course, but also host microbiome interactions. Animals and humans were never in evolution alone. So the nervous system, when it evolved, had to control this microbial colonization. The partnership is in danger because we use lots of antibiotics, and these antibiotics kill, reduce, or disturb these microbes. And the antibiotic use is increasing. Again, here a statistic from the World Economic Forum, and you can see, I mean, Turkey and Tunisia, but also many other United States, Luxembourg, European countries, we just have too much antibiotics in our food, also medical descriptions, of course, but particularly in the food, in agriculture, in animal breeding, and these antibiotics really are bad because, as I told you, some bacteria are anti-inflammatory and actually help wound healing. For example, this Akkermansia bacterium here sits in our gut and it helps wound healing. But if we treat people with antibiotics, this interferes with this bacteria, it kills them, and other bacteria can grow, and then it causes chronic inflammatory disease. Because when you remove some of the bacteria, other bacteria have an open niche, an ecological habitat, and they can actually settle there. We call that colonization resistance because the normal microbiome prevents that pathogens can really come. But if you remove these normal microbes, pathogens can come. One of the leading experts worldwide is Dr. Martin Blaser. He has written a book which is now translated in 17 different languages, Missing Microbes. And Martin studies in depth the functional consequences of antibiotics on health consequences in humans and in mice in context of the microbiome. So when you followed all that, you may ask, what can I do that a healthy microbiome is still there? And I told you about fecal microbial transplantation. That is not a good thing. That medical doctors do at certain cases in certain patients when nothing else helps anymore. And we are working on a better way to transfer microbial products into a patient than by fecal microbial transplantation. But this works, as I said. The other major point where you can manipulate and help and support your microbiome is in the diet, having a normal, not processed, plant-rich diet, which our predecessors 70 years ago, they had no refrigerators and they didn't process the food, and they didn't go to McDonald's, but they had to cook by themselves, and they cooked what was there and what was growing on the fields. That is what evolution has made our gut and our intestine for to product. And the future in this field, of course, is also probiotics. At the moment, there is not much really known on the functional interaction between probiotics and the normal microbiome. Many people are working on that, and certainly a promising future field is the development of probiotics. That's what you can do. And very easy, you can allow your kids access to nature. That is nothing really dangerous. And my colleague and friend, Dr. Brett Finlay from Vancouver, has just published, he's a children doctor, has just published a book, Let Them Eat Dirt. We were all used in our childhood that we go out for nature and that we get microbes from the environment. And some of them then colonize our body and we need them. But our modern world, our urban world in our mega cities has not much possibilities for children to go out and get exposed to microbes. That brings me to the very last point of this presentation, hygiene measures. What happens in these last 16 months when we have to follow strict hygiene conditions and we have to follow them? Social distancing and taking care and wearing a mask and vaccination. But that comes also with a cost. And while normally, as I just told you, the bacteria from the environment, from the family, vaginal delivery, the diet, adds up, and so we have a diverse microbiome as a child, as an adult, as an old people, and we lose this with increasing age again. That is the normal way of how to get and how to lose microbes in our gut and our skin. However, during this pandemic now and all the measures, what we have to do, the gain of the microbes from the environment is getting very difficult because families keep their food pretty much sterile, social distancing, kids cannot go out so much in the community, and they cannot increase their rate of the microbes. And on the other side, normally we lose microbes anyway, but with these new pandemic hygiene measures, this rate of loss of diversity of the microbiome is increased dramatically. Hygiene, antibiotics, social distancing, and all these kind of reactions. So what we really want to tell you is that hygiene measures are necessary, and we only get rid of this virus by vaccination and by following the rules. But in evolution, we were grown up and developed and evolved in a microbial world. And as I just showed you the last 20 minutes, microbes have a major impact on our health and our fitness. So any measure in the COVID-19 response comes at costs. And we have to carefully balance what to do, and something are really not necessary. So then please don't do it, because it may even more damage the microbiome. I summarize my presentation, and I hope I have shown you that health and fitness of animals, including humans, is fundamentally dependent on multiple organisms of several species. By removing microbes, antibiotics, hygiene, our bodies don't function normally anymore. We get sick. And that we have now to consider organisms as meta-organisms. And the modern medicine must return to a holistic view of organisms and the patients. Only the application of a reductionistic approach, go very deep in a very single tissue, can of course go to the functional interactions, but you need the holistic view to understand the complexity. So please think ecosystems, organisms, and the patients from a perspective of a biological association that has a long evolutionary history. In a book I have written some years ago, the last sentence was, "The time has come for a holistic understanding of complex life processes, and the time is here, and I invite you to participate in that time." With that, I'm at the end of my presentation, and I acknowledge my funding sources. It's particularly the German Science Foundation and the Canadian Institute of Advanced Research. Thank you very much for your attention.